Good afternoon, I'm Brianna, and today I will be talking with you about decreased plasma comp and increased plasma CTX2 levels in a Chinese pseudo-achondroplasia family with novel comp mutation. Here are the authors here. I chose this paper because the mutation that causes pseudo-achondroplasia has always been an interest of mine. So when I came across this paper, I decided to read and learn more about it. So pretty much this paper is going to look at the comp, which is a protein, and how it affects the mutation and results in pseudoachondroplasia. So some background information you need to know before I get into the gist of the paper, one of which is the pseudoachondroplasia, which in my paper and throughout this presentation will be known as the PSACH. It's an inherited bone growth disorder, also commonly known in today's society as a form of dwarfism. It's an autosomal dominant pattern. And so what it is a mutation in the comp gene, which I'll talk about in later slides. So characteristics of this mutation, it results in brachydactyly, a short stature, scoliosis, which can be seen here in this image, which is the curvature of the spine. The people with this mutation will also deal with a lot of joint pain. So here you can also see they have short and smaller hands and arms, and you also see that their fingers are more cone-shaped. They have curvature in the legs, which is not more normal within those without the mutation, and they also have issues within their hip bones. So this condition is recognized between ages 2 and 3. It's not something that can just be known as soon as a child's born unless you do blood tests. So as I said, pseudoachondroplasia, it's an autosomal dominant inheritance. So this is when there's the presence of one mutant gene as well as one wild type gene. So within this type of inheritance, the mutant phenotype is resulted in just the presence of one mutant gene. So the mutant is dominant over the wild type allele, which in this you can see that the mother has 2 normal genes here, while the dad has 1 mutant gene. And within their kids, just that presence of 1 mutant gene results in these children having the condition, whereas these don't because they have 2 normal wild type alleles. Another aspect is the cartilage oligomeric matrix protein, also known as COMP. So this is a very essential part of cartilage development. So this is found in the chondrocytes, and what they do is they secrete the cartilage and then are embedded within them. So within the pseudoachondroplasia mutation, which is found here on chromosome 19 at 13.1, it builds up and results in the cell death, thus preventing bone growth, and that's why those with achondroplasia have a shorter stature. So the other big aspect is what exactly is cartilage. So cartilage is a tough, flexible tissue, and it is what covers and protects the ends of your bones. And in this paper, they also focus on type 2 collagen, which is linked with cartilage, and the COMP protein. So as you can see here, this is collagen type 2, and this is the COMP protein right here. So what the type 2 collagen does, also known as the CTX2, it's a basis for articular and hyaline cartilage, and this is what makes up 50% of protein within your cartilage. So one of the methods they used to find their results within this paper was they transfected HEK 293 cells. So HEK 293 cells are human embryonic kidney 293 cells, and they are used commonly in research because they have reliable growth, and they're also very good for transfecting. So here I show HEK 293 cells. So what they did in this particular study is that they plated them for 24 hours before the transfection, and then they transfected them with 2 micrograms of either the pcDNA 3.1 wild type comp, the pcDNA 3.1 mutant comp, or pcDNA 3.1 MYC empty plasmid. So after this, they went through a series of incubations in order to be ready for the rest of the research needed. Another method they used within this paper was Western blot. So a Western blot is the detection and analysis of a specific protein, and these proteins are determined because they are separated by their molecular weights. And you can see that you have your sample preparation here. They undergo gel electrophoresis, and then they also go through processes that denature them, and the primary and secondary antibodies are added, and then through detection, you then get a gel that allows you to see which proteins are present by the bands and the molecular weight at which they fall. So for this paper, they said they used the achondroplasia family. So here's the pedigree. So you can see that within their family, they both have people with and without this condition. So they specifically focused on this program here. It's number 3, and it's indicated by the arrow. And then the other person within the family they really focused on was the pre-symptomatic carrier. So it is his daughter here, indicated by this non-filled in circle, but with that vertical line through it. So what they found is that the pre-symptomatic carrier showed no discrepancies in either comp or CTX2 levels compared to the controls. So they used age-matched controls when they did this, and what they found was the PSACH patients compared to their controls in age ranges of about 34 to 65, they showed a significantly lower value of the comp and a significantly higher level within the CTX2. Whereas for the program's daughter, so the pre-symptomatic patient, there was no mean difference in either the comp or the CTX2. So this was also shown in these graphs here from that table. So you can see here that in the comp levels, you have the pseudoachondroplasia patients here with lower levels of the comp compared to the control, whereas in the pre-symptomatic carrier and the control, they're relatively the same. And then over here in the table depicting the levels of CTX2, you have higher levels of the pseudoachondroplasia patients compared to that of their control. And while you see a slight rise in the pre-symptomatic carrier, they still are relatively the same. So they also saw the expression of wild type and mutant comp. So this was through the Western blot, and they used anti-MYC. So there was the expression of both the wild type and mutant proteins within the HEK293 cells. You can see this here with both the variants present at both 12 and 24 hours post-transfection. But you can also see that the mutant showed a higher quantity compared to the wild type. So this is again seen, so you have the immunofluorescence of these 24-hour post-transfected cells. So the wild type, which you see here, you see that it is expressed in the cytoplasm compared to where in the mutant, the accumulation was within the cell membrane. So overall, they found that there was a decreased comp and increased CTX2 may pair as a diagnostic marker for pseudoachondroplasia patients, but it isn't applicable for the asymptomatic carriers as seen because there wasn't varied in the mean levels compared to their control. So for future research, it would be important to demonstrate if the extracellular mechanisms contribute to the computation causing this pseudo-achondroplasia. And these are my references. Thank you.